Okay, now we studied the following important points with regard to DNA. One is Frederick Mischer, who discovered a chemical substance in DNA, chemical substance called nuclein in the pus cells. Second point is Watson and Crick, who gave the model of DNA. Third point is there are two DNA strands which run anti-parallelly to each other. Why it is anti-parallelly? Because one runs in 3', 5' direction and another runs in 5', 3' direction. So it is anti-parallel movement. And the backbone of the DNA is made up of sugar and phosphate. The backbone of the DNA is made up of sugar and phosphate. And the fifth point is nitrogen bases are always attached to pentose sugar of backbone. Nitrogen bases are always attached to the pentose sugar of the backbone. And next point, always there is a particular method in attachment of these nitrogen bases. How it will be? Adenine always forms a double bond between thymine. Adenine pairs with thymine. That means purine always pairs with pyrimidine. Purine always pairs with pyrimidine. And that is adenine of the DNA pairs with thymine of DNA with 2 hydrogen bond. Then guanine of DNA pairs with cytosine of DNA with the 3 hydrogen bond. 3 hydrogen bond. Guanine of DNA pairs with cytosine of DNA with 3 hydrogen bond. So that you should remember. Always adenine pairs with thymine, guanine pairs with cytosine. There is no base pairing between adenine and cytosine, guanine and thymine. And always the base pairing. So what is the next point that we should remember? That is sixth point, isn't it? Sixth point that we should write. Base pairing of nitrogen bases occur in a particular form. That is, adenine pairs with thymine with 2 hydrogen bond and guanine pairs with cytosine with 3 hydrogen bond. Guanine pairs with cytosine with 3 hydrogen bond. Then seventh point, the two strands, the nitrogen bases of the two strands are complementary to each other. Complementary to each other. What do you mean by complementary? Puraka. Complementary means puraka. That means, for example, if we know the nitrogen base sequence of one strand, that is adenine, adenine, thymine, guanine, cytosine, adenine, cytosine, guanine. If we know the base sequence of one strand, we can write the base sequence of another strand. So, in front of adenine always there is a double bond, and what should be there? Thymine. So, here also thymine. Here, so thymine pairs with adenine. Guanine pairs with 3 hydrogen bond, cytosine. Cytosine pairs with guanine with how many hydrogen bond? 3 hydrogen bond. Adenine pairs with thymine. Cytosine pairs with guanine. Cytosine pairs with guanine. Guanine pairs with cytosine. Like this it goes. That means one strand, if we know the nitrogen base sequence, we can analyze the base sequence of another strand, because the two strands are complementary to each other. That is the 7th point. And what is the distance between the nitrogen bases? That is 3.4 angstrom. The distance between the base sequences are about 3.4 angstrom. So that is the next point. 8th point. The distance between adjacent bases will be 3.4 angstroms. And for one complete turn, DNA takes around 10 bases. How many bases for one complete turn DNA takes? 10 bases, base pairs. That means for one complete turn, I'll write it properly. Now you can analyze. For one complete turn, from here to here, it is one complete turn. You can see 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. For one complete turn, it requires, so how many base pairs? If you can't understand this, I will write the base sequences only. Because for the first time, yes. Guanine, cytosine, 3 hydrogen bond. Thymine, adenine, 2 hydrogen bond. Thymine, adenine, 2 hydrogen bond. Adenine, thymine, 2 hydrogen bond. Guanine, cytosine, 3 hydrogen bond. Cytosine, guanine, 3 hydrogen bond. Adenine, thymine, 2 hydrogen bond. Guanine, cytosine, 3 hydrogen bond. Look here. How many bases are here? Base pairs? 10 base pairs, isn't it? So, for one complete turn, it requires 10 bases into the distance between the adjacent bases is 3.4 angstrom. For one complete turn, it takes about 34 angstrom. How much it is? 34 angstrom. And the width of the DNA is 20 angstrom. The width of the DNA is 20 angstrom. So, for one complete turn, 9th point. For one complete turn, it requires 10 bases, base pairs, 10 base pairs. For one complete turn, it requires 10 base pairs. So that means for one complete turn, it requires 10 base pairs. That means what? That means for one complete turn, it requires 10 base pairs. So for one complete turn, it requires 10 base pairs. The total distance is 34 angstroms. 34 angstroms. For one complete turn, how much distance is required? It is 34 angstrom. Then the width of the DNA, is 20 angstrom. 20 angstrom. The width of the DNA is 20 angstrom. Next, 12th point, in between adenine and thymine, there is 2 hydrogen bond. 2 hydrogen bond, and between guanine and cytosine, there exist 3 hydrogen bond. 3 hydrogen bond. There exists 3 hydrogen bond. So in between adenine and thymine, there exists 2 hydrogen bond, and in between guanine and cytosine, 3 hydrogen bond. So always purines pairs with pyrimidine. This point you should write. Always purine pairs with pyrimidine. So this helps to maintain the constant width. This helps to maintain the constant width. Constant width. This helps to maintain the constant width. This helps to maintain the constant width. Next, Erwin-Chargaff rule. They may ask it separately for 2 mark. Erwin-Chargaff rule. What does rule says? Erwin-Chargaff's rule. What does rule says? It says that in DNA, the proportion of adenine is equal to the proportion of thymine, and the proportion of guanine is equal to proportion of cytosine. It says that in DNA, listen it carefully, in DNA, the proportion of adenine is equal to proportion of thymine. Proportion of thymine. And the proportion of and the proportion of guanine is equal to the proportion of cytosine. Correct? Because it is true. Because how many adenines are there in a DNA strand? Same amount of thymine should be there because it should pair. Adenine should pair. There will not be any unpaired bases inside the DNA. Every base should be paired unless there is a mutation. So they should be paired, isn't it? So when they should be paired, then automatically whatever the number of adenine is there, the number of thymine should be same. If there are 100 adenine in one DNA strand, what should be the number of thymine? 100. If there are 99 adenine in one DNA strand or 99 thymine in one DNA strand, what should be the number of adenine? 99 only. It will not be 100. And the number of guanine in the DNA strand is always equal to the number of cytosine, or number of cytosine in the DNA strand is always equal to the number of guanine. If number of guanine in the DNA strand is 50, number of cytosine also should be 50 only. It should not be 51, because there is no unpaired, I'm telling, there will not be any unpaired bases in the DNA strand. There will not be any unpaired bases in the DNA strand. That you should remember. So this is what the Chargaff identified and told, hence it is called Erwin-Chargaff rule. Then what it says, that is adenine plus cytosine number is always equal to thymine plus guanine number. Correct? So the total number of adenine and cytosine is equal to the total number of thymine and guanine. Or I can write it like this also. I can write it like this also. Dear students, this chapter is very important for your NEET point of view. So if you have any doubt, please call me and ask the doubt, okay? So, or we can write this as adenine plus guanine is equal to, or thymine plus cytosine is equal to 1. Is equal to 1. So, the number of base pairs in human beings, number of base pairs in humans, it is 6.6 into 10 to the power 9. Hence the length of the DNA will be 6.6 into 10 to the power 9 into 0.34 into 10 to the power minus 9. That is 2.2 meter. What is the length of the DNA in human cell? It is 2.2 meter. In E. coli, it's a bacteria, length of DNA is equal to 1.36 millimeter. That is 1.36 into 10 to the power minus 3 meter. 1.36 into 10 to the power minus 3 meter. Then the other things we should remember is the number of base pairs is equal to 1.36 into 10 to the power minus 3 divided by 0.34 into 10 to the power minus 9. That's equal to 4 into 10 to the power 6 base pairs. So this is about the nitrogen bases, the length of the DNA in E. coli. You remember only this one. That's enough. 2.2 meter in human and 4 into 10 to the power 6 base pair in E. coli. Okay. Now, some other aspects which may help your competitive exam. Psi 174, a bacteriophage. It's a bacteriophage. Means virus has 5386 nucleotides. Has 5386 nucleotides. Then bacteriophage lambda has 48,502 base pairs. E. coli has 4.6 into 10 to the power 6 base pairs. The haploid content of human. Haploid content of human DNA, that is what 3.3 into 10 to the power 9 base pairs. These are all, you should keep it in your memory. That Psi 174 is a bacteriophage. It's having 5,386 nucleotides. Bacteriophage lambda has 48,502 base pairs. E. coli has 4.6 into 10 to the power 6 base pair. Haploid content of human DNA 3.3 into 10 to the power 9 base pairs. So these are all the things that you should remember.